This next video is to do with the polymerase chain reaction. So I wanted to just run through with you what DNA polymerase was first. So we understood that polymerase means using, it's an enzyme to create a polymer. So polymerase chain reaction is using that enzyme to create polymers in a chain reaction, which means that one thing leads onto another, leads onto another, leads onto another. So polymerase chain reaction is using that enzyme DNA polymerase to create many, many copies of a particular strand of DNA. So we often consider this to be one of the most important advances in biotechnology that we've made since we started playing around with DNA. So DNA was only discovered about 50 odd years ago. And then since then, we've been trying to crack the code. So I will link you to some videos on CRISPR. In particular, there's one really cool one, and hopefully we've got some time to watch it in class and discuss it. You guys just missed out. The next study design is going to be talking about CRISPR. But for now, we're just going to talk about polymerase chain reaction and why it's so important, and why have you never heard about it if it is so important. This is really vital because polymerase chain reaction allows us to copy from a tiny sample of DNA, copy it millions and millions of times. So prior to the invention of polymerase chain reaction, when we found DNA and we wanted to manipulate it, we would only have the sample that we extracted from the source. Now, that is problematic because once you've used it up, it's all gone. And so you only have a limited number of experiments that you can perform on it. So PCR gives us this ability to take that very small sample, multiply it as many times. So essentially we're just making photocopies of the DNA. Then we can do this experiment with it, we can do that experiment with it, we can do this experiment with it. So we used, the best example of why this is important is in a court case like O.J. Simpson. So O.J. Simpson was tried for the murder of, hang on. Sorry, I wanted to get her name correct. So he was tried for the murder of his wife and a friend of hers. So when they did that trial, it was one of the first trials that they used DNA evidence. The problem was that after they'd done the analysis on the DNA, they actually used up all of the sample. So with these old technologies, they weren't very reliable, they weren't tested enough yet, and they used all the DNA. If they had used PCR prior to that, we could have made copies of that DNA. And now, even now, in 2020, and this was back in 1990, oh goodness, now I'm testing myself on all of these things. 1994, that he was first tried. If we had that evidence still, we'd be able to retest it and see what we could find out, which is much more than we used to be able to. So PCR is really valuable in not destroying all evidence of a sample, and therefore you can keep some frozen if you need to test it later on. On a side note, Netflix have a really good dramatization of the trial called The People versus O.J. Simpson. It stars a whole bunch of, Cuba Gooding Jr., I think, plays O.J. Simpson. It's a really interesting story if you want to read about it and watch it if you haven't heard of who he is. So there are some of the applications of that. So I'm just going to share this because it seems to have stopped mirroring. Let's try again. There we go. So it's possible to generate millions and millions of copies. So you can use trace DNA from crime scenes. Also things like fossils. You can find fossil DNA and you don't want to destroy that in the first time you use it. So you can use PCR to do that. You can also use it on extinct animals and plants. And also you want us to have sufficient quantities. Sometimes you only find things in trace amounts and you need to be able to get sufficient quantities. So let's have a look at the components, what we need for PCR. So the first thing we need is going to be our DNA fragment, the thing that we want to copy. All right, so we're going to have this DNA fragment that we want to copy. We're also going to need to add DNA polymerase. In particular, we call the DNA polymerase that we use Taq polymerase. Now, the reason I'm telling you what it's called is you're fine to just write DNA polymerase, but the technical one is Taq polymerase. It's because it came from a bacterium. It's very, very tolerant to high heat. So this bacteria, we call them thermophilic bacteria. So we extracted this DNA polymerase from these thermophilic bacteria. And it's very important that these polymerase molecules are tolerant to high heat, and you'll see why very shortly. We also put in a bunch of primers. Now, what a primer is a short sequence of nucleotides. So imagine there's your sugar phosphate backbone. So there's your sugar phosphate backbone. Then in addition to your sugar phosphate backbone, here are your nucleotides. So what we do is we know we have a whole bunch of different primers for different things, and we know that these are going to attach on one end of the DNA. Essentially, this is the starting point for PCR to occur from, and you'll see that in the pictures in a second. If we're going to be copying DNA, what do we need to put in the soup as well? We need to put a bunch of free DNA nucleotides. And then all of these things we put in a special machine called a Thermo Cycler. So the Thermo Cycler can be as small as this. Now, a little box, you put all of these things in tiny little tubes, and what the Thermo Cycler does is it cycles through different thermal levels. So you can go hot, then cold, then medium, then hot, then cold, then medium. And that's going to make sense when we go through this process. So to begin with, and I'm just going to get rid of that up there, here we have our target DNA sequence. So when we get our target DNA sequence, plus we add our primers, plus we add our Taq polymerase, plus we add our free nucleotides. So all of these things are put in a thermocycler. And we start to go through the process. Now, there are 3 stages that we need to understand about PCR. So we are now on page 6, and we are looking at the 3 stages. For each stage, I want you to be able to describe what happens. So remember, description is simply what happens, and then explain why those things are happening. So what we have here is this is showing us stage 1, stage 2, and stage 3, and I'm going to break them all down for you. And then you would do that cycle once, twice, multiple times. So here's another picture of it. This is the one we're going to break down. So we're going to look at stage number 1, stage number 2, and stage number 3. Here is stage number 1. So in stage number 1, you don't need to know that it's called denaturation. If you want to write that down, you can, but the names are not important. What is important is that you understand the temperatures and what is happening at each stage. So during stage number 1, it is the hottest stage. We increase our temperature to around 95 degrees Celsius. You can see here's a diagram that says 94 to 96. And what that's going to do is it means that your green DNA sequence in the middle here is going to split. The hydrogen bonds in the middle can't withstand that kind of energy in the mix. So what they do is they break apart. So now you've got 2 strands of DNA. So you've gone from your double strand of DNA to 2 strands here. And that's because you heated it up. So it broke the hydrogen bonds in between the strands, and the strands separated. So the description is that you heat them up in the you put everything in and you heat it up in the thermocycler. The explanation is the high temperature will split apart the 2 strands. Now we've got these split apart strands, we need to cool them down again, because nothing is going to stick. You won't be able to stick anything at those kinds of high temperatures. So when you cool them down again in stage 2, the primers will join, or anneal is the word, to the end of the DNA fragments. So one will attach to one 3' end, the other one will attach to the other 3' end. Doesn't matter about 3', 5', but they are just attaching to the ends. Now what these are doing, these are providing starting points for the next stage. You have to cool it down because otherwise they won't be able to form the hydrogen bonds that are in the middle here. Yeah, so the hydrogen bonds, they'll come like this. The other good thing about this is so if you've got your 2 strands of DNA, if you clamp something onto the end here, this one can't rejoin. So that's a good thing. So by having the primers, you can't get those strands rejoining. The final stage is when we go to a medium temperature. So we started off, stage 1 was high, stage 2 was low, around 55 degrees, and stage 3 is in the middle, around 70, 72 degrees. Now what happens here is this is where Taq or DNA polymerase binds to the primer and moves down the strand. So it's moving in the blue direction here. Yeah, and it's happening on the other strand too. Moves down the strand and joins free nucleotides. So here are the free nucleotides being joined. Free nucleotides joined, and now we end up with 2 lovely double-stranded pieces of DNA. Why do we use that temperature? Because that temperature is the ideal temperature for Taq polymerase. Why don't we use human polymerase? Think about it. What was the coldest temperature we got to? 55 degrees Celsius. 55 degrees Celsius is way hotter than something that would exist in the human body. So we don't want to use human polymerase because it's going to, what? Denature, because it's an enzyme at these kinds of hot temperatures. So by using the Taq polymerase, or by using the bacterial polymerase, we end up with this protein that can deal with the temperatures between 55 and 90 degrees Celsius. So they're your 3 stages. So you can see here, number 1, you've got your splitting of the strands. Number 2, you've got the primers sticking. Now I'm doing this very brief. And number 3, you get your elongation or new strands forming. Now the benefit about chain reactions is that each one of these new strands then acts as the template for the next one. So after one replication, you start off with 1 piece of DNA, you end up with 2 pieces of DNA. After the next stage, you're going to end up with 4 pieces of DNA. After the next stage, you're going to end up with, doubling again, 8 pieces of DNA. So what you get here is you get exponential growth every time you do a cycle of PCR. And here are some more pictures. So these pictures are in your notes. This one is really nice to show you that it is a cycle and that it just keeps going around and around. So you start off here with your DNA fragment, you heat it up and it breaks into 2 bits. Then at the second stage, you get the DNA primers binding to the end. They stick at a lower temperature. Then you heat it up a little bit more. So you go from here to here to here. And this is where your DNA polymerase is going to attach your free nucleotides and you end up with 2 new DNA fragments. Then you heat it up again and you can go through this cycle over and over and over again. And that's how PCR works.